My name is Noah Ashley. I'm an assistant professor at the biology department here at Western Kentucky University, and my field of study is really two parts. One is looking at the ecology of sleep, and also the other is looking at immune sleep interactions in mice. So the grant that I just received is from the NIH, and it is a grant that really is looking at how sleep loss affects the physiology of an organism and also the immune system of an organism. So we know that when individuals lose sleep, when they undergo either sleep disturbance or they pull an all-nighter, we know that the immune system actually changes. And I'm really interested in those changes. My lab is really focused on trying to model a certain type of sleep abnormality, and that is obstructive sleep apnea. So that's a condition where you have individuals, when they are sleeping, they tend to go through periods of apnea, which is basically they lose their breath, they have a lower oxygen saturation, and that leads them to waking up during the night. And so we actually model that in mice. One of the main processes that happen is that there's an increase in inflammation, not only in the periphery, like in different target tissues in the periphery, but also in the brain. So we have a number of different techniques that we use. One is to look at certain molecules of the immune system that are elevated during inflammation. So there's interleukin-1, tumor necrosis factor, interleukin-6. We will look at the gene expression of those particular cytokines and see if they're upregulated during sleep fragmentation. And so this grant really looks at how that inflammatory response develops and whether or not certain hormones are involved in its regulation, so that there's an increase in stress hormones and also an increase in what are called catecholamines. And we know that they're altered during this period of sleep loss, but we don't know how they affect inflammation. So the goal of this project is to look at the effects of catecholamines and its downstream effects, being an increase in blood pressure on this inflammatory phenotype that's created in mice that have been exposed to sleep fragmentation. And feel very fortunate to get this RF-15 funding. Doing any type of molecular work or neuroscience research is pretty expensive because it requires expensive reagents. Also, it's going to help me hire a postdoctoral associate that will specialize in neuroscience. In addition, we have some funding for a new cryostat that is able to slice tissue sections, basically a couple different microns in thickness. And then we actually look at that tissue underneath the microscope and look for different cell types, like microglia is one cell type that I'm interested in looking at. I would not have been able to be competitive for this grant unless I was able to get seed funding from KBRN to collect the preliminary data that was necessary to show that I can perform these experiments that I outlined in this new grant. So yeah, I think that it's an incredible resource that we have here for the state of Kentucky.